Yeah, welcome everyone for joining today's GHGA lecture series. It's a really great pleasure today to welcome Helen Parkinson, who's joining us from the European Bioinformatics Institute. It's great to see via distance a former colleague. The virtual world of not being able to visit has made it easier to at least meet in this forum, but I really also hope, Helen, that we can have you over here in person and at some point in Heidelberg. So Helen is head of the molecular archival resource at the European Bioinformatics Institute, which is one of the major pillars of molecular data sharing. So primarily incorporates the European Nuclear Archive, so that's open access genome sharing, but also controlled access genome sharing, the EGA, and that's obviously very, very deeply of interest to the German Human Genome-Phenome Archive as a federated node that is part of the EGA. So this is a very, very topical honor to have you here and have you speak to us today to hear more about the work from the archival side. A few words about Helen. So Helen is a geneticist by training, so actually has done a postdoc in Leicester and before that a PhD thesis in the genetics field, and now shifted to bioinformatics and the computation side of things back in the 1999, so with the computation side for quite some time. And Helen describes her passion as making semantic data integration work and providing users with useful data. And that's obviously something we're also trying to achieve. And we're really just starting here on GHGA. So we really, really look forward to hearing more about how you have already made that happen. And yeah, with that, I think I'll hand over to you, Helen. Thanks again for joining us, and the stage is yours.
Thank you. So somebody has to stop screen sharing before I can, I think.
Shall I go over the housekeeping maybe quickly?
I'll be quiet for a moment.
Well, now it's gone again. Okay, this is what happens.
Sorry, yes, maybe you can, Oliver, maybe we could mention that the next GHGA lecture, which will take place on January 24, will be, we will have we have invited Joachim Schulze, and he will talk about swarm learning from research to medical use of genomic data. And some housekeeping for the people who already assisted the last time, they already know it, that all participants are muted, that questions and answers will be all at the end, but you can post your questions in the chat, and Oliver and I will moderate this at the end. And yes, and that you have already realized that this lecture will be recorded. So now, yes, please, Helen, you can start. Thank you.
Perfect. Thank you very much for the introduction. I'm really happy to be here. It's an honor to follow Ivo Good, who is a distinguished scientist and former collaborator. And in a minute, my slides will come. Okay, does it look good? If someone could nod.
We can see your slides, yes.
Perfect. Thank you very much. So I'm going to talk about data integration, also a little bit about interoperability for biomedicine. As Ollie said, I have been head of the molecular archives, but actually my group works on a lot of different things, and we do a lot of making data integration work for different scenarios. And so I'll talk through some of those and some of the strategies we use with some examples, and I'll be really happy to take questions at the end. So in this talk, I'll talk a bit about the need for data integration, some of the complexity, and some of the federation that Ollie spoke about at the beginning. I'll talk about solutions and example scenarios, and I have a diversity of those. I'll talk about interoperability, enabling integration, and some of the outlook and where I think the world is going, and some of the things I think will be challenging. Okay, so I work at the European Bioinformatics Institute. We're an outstation of EMBL. And I need to just move the people because they're obscuring my slides. There we go. And so we do lots of different things, including research, and I collaborated with Ollie in his research context when he was at Kingston, but also training, industry engagement, and coordination of bioinformatics. And actually the coordination piece is really important for this, because if we don't have the pieces that bring together the data for integration, it becomes really challenging. I work on the scientific services side of EBI, and I will mainly talk about that today. mouse on the slide. There we go. So we're really dealing with a lot of large data sets, and there's a very large demand. So just to give you some idea of the stresses and things that really we have to deal with, is 82 million requests to EBI websites every day, from 41 million unique IP addresses, 500,000 people or more addressing the train online resources, so how we do our training, and 390 petabytes of raw storage. And this really becomes a challenge to actually scale anything in this space. And even for smaller databases, it becomes challenging to scale. And I'll talk about why that's important for integration in a bit. So we have more than 40 open data resources, and I will talk about 3 today. 9 of those are data deposition resources, and many of them are knowledge bases. And so there are lots of drivers for integration, from how we move from data to information to knowledge, to application of that in context. And that application might be screening, it might be in the context of a health system, or it might be in the context of drug discovery. If you're interested in what we've been doing in the last year, there's a paper which has just been published which describes some of this. But one of the joys of working in this space and dealing with data integration at this scale is there's so much variation in how people want to use the data. And I think using the data is the thing that I think about most. And so this is an image from our annual report. And it's a representation of how the data is shared between resources. And so each piece of color on this wheel represents a resource, and then the edges represent the way that one resource interacts with all the others. And you can see some things are really highly connected. So for sequences, we have data from the nucleotide archive going to Ensembl and Ensembl genomes, but also into UniProt, because all of the translated proteins need a genome sequence underneath. And so we have a massive amount of data flow and dependency to make the connections across these edges work. And that becomes really challenging. And it becomes really challenging when we actually get it closer to users to understand how people want to use the data. And that's what I'll talk through today. So I want to talk a bit about the landscape we work in, and a bit about the Global Alliance for Genomics and Health before I start getting into examples. So in 2017, the Global Alliance for Genomics and Health was starting to talk about genomics entering healthcare. But I think what we can see in 2021 is that genomics has really entered healthcare. And I've put a couple of examples on here. So this is a GA for GH slide back from 2017. And we were talking about Genomics England in the UK, sequencing 100,000 rare disease patients and diagnosing them. But actually, that 100,000 is now morphed into the NHS Genomic Medicine Service, which operates for the benefit of the whole of the UK. And that's 66 million people and all of the individuals who might need to get referred to the Genomic Medicine Service. And that's a clinical service. In 2021, there's a vision to sequence hundreds of thousands of newborns, because we know that many of those newborns are going to end up in neonatal intensive care, and many of them will have undiagnosed diseases. And actually the diagnosis piece is important for their families, but also has a huge health economic burden if they're undiagnosed, and outcomes are much better from early diagnoses. And in the US, the All of Us program, which is a bit like UK Biobank, or Cora in Germany, it's recruiting more than 1 million participants, but it's already 440,000 people in, and a third of 1 million people have given samples. So the scale is really growing. And this really changes how we think about data integration. So why integrate data? So in preparing for this talk, I went back and I did a bit of reading and thought about some meetings I've been to quite a long time ago. So I've got 2 reference papers here, one from Norman Payton from the University of Manchester, and Carol Goebel and Robert Stevens, also from Manchester, who talked about the strategies for data integration back in the early 2000s. But I think the desire is still the same, which is to enable data set exploration, analysis, and exploitation, to deliver new knowledge, and in doing that, to test or confirm our hypotheses. But the way we do it is a bit different. And so the world has changed a bit because of that landscape, particularly when we're working with human data. And I'm going to talk about human data today, although I work with flies and mice. And as a model organism biologist, I'm still very interested in other species and what they tell us about human biology. So it's worth going back to think about why data integration in biomedicine is complex. So firstly, the scale is enormous. In my lifetime at EBI, I've gone from an institute of 180 people to more than 800. And we're not there because we're all doing things in the same way. It's because the data has grown, and there are many more data types as we've gone through these kind of paradigm changes in data generation. The data are becoming more complex, They're both vertically complex in terms of the granularity, but they're also horizontally complex because the data are fragmented and the data exist in lots of different resources, and we're spending a lot of time trying to figure out how to federate. The quality of the data become challenging when they're being generated at this scale, and also how to knit them together in meaningful ways, so data readiness. The governance and licensing is very challenging, and so we're dealing with federation, but also the path that gets us from data to knowledge. And on the bottom of this slide, I've indicated some data growth from EBI data resources. It's from the paper I mentioned before about the review of the last year. But I've also got a slide from the Open Targets platform. So Open Targets is a pre-competitive public industry collaboration. It was started with GSK, but now it includes several other pharmaceutical companies. And this is a nice example of BMPR2. So I was involved in the positional cloning of this gene in 1999. It's associated with primary pulmonary hypertension, so a rare genetic disease. But actually, to understand about how you might develop drugs for primary pulmonary hypertension, you need to know a lot of things. Is there drug data? Is there mouse phenotype data? Is there pathway data? Is there expression data? And you can see lots of linked data sets here. So I'll talk a little bit about how data sets get integrated for different applications and what kind of things we need to do to deliver those. But this is the problem. And we've got a couple of examples here of how things need to be integrated to do things, data, to analyze or to test hypotheses. But that raises the question, what does good look like? And this is what I think good looks like in terms of the experience with the data resources we work on. So those data resources should be scalable, extensible, modular, and service-based. And so I live in a world where I'm judged by, can the 200,000 unique users a year of the GWAS catalog access the data in real time? And so we need it to be service-based because we have a contract with the user. Is it simple for the user? Are the user interfaces and the programming interfaces usable for the things they want to do? And are we interoperable? Because we know we're not the only resource that people will use, but we also know internally our data must flow up through those resources that I showed. They should be plug and play because we want to put things together for different scenarios. For example, when we operate data resources in different jurisdictions. The quality should be aligned to the use that we envisage for the data, but we should also be able to go back and upgrade the quality if we need to. And we need clear licensing and evolving governance models. And I think the governance models become one of the most challenging things. Before I existed in a world about knowing about large molecular data sets, I never cared about governance or licensing. Who owned the data and what they wanted to do with it was not a concern of mine because I worked in my world of my data and I knew what the consent was. But as soon as you want to aggregate data, it becomes challenging. And I'm interested in the data to knowledge path. So how we get from a very large database to a knowledge base that tells us something about perhaps a gene or a variant or a disease, but also how we get back from knowledge to data so that we can understand what the provenance model is, to know that we're really dealing with something trustworthy, but also knowing when the next data set comes along and disrupts our knowledge, that we will be able to cope with that. All of this sits in the context of the findable, accessible, interoperable and reusable principles, the FAIR principles published in 2016. And I'm going to focus a bit on interoperability for integration in the rest of this presentation, because much of what we do would not be possible without this. But we also absolutely need the data to be findable and usable, and that comes back into the governance piece. And the reuse, I think I'm going to demonstrate, is how we mash things up for different contexts, but using the same data in different ways. So what's our approach? So I work in a world where I deal with high volumes of data and deliver services, but actually we also have to think about people and value. So my take on the data and services piece is that I want to build on a core service stack and use common technologies, because I don't need, my team's about 30 people, I don't need everyone to be using a different technology. That I want standards, formats, semantics, provenance, and identifiers all to be in place and to be usable, so to correspond to the FAIR principles. I care about governance and compliance, particularly when we're dealing with human data, but not only human data. The Kyoto protocols determine how we are compliant in other contexts, for example, for data which might have access and benefits restrictions. I also care about how we sustain things, and it's important to understand when to decommission and deprecate and to know when to build something new. The people I think about are our users and how they engage with us through user experience, so testing things, trying to find out what they want to do. There are collaborators. None of this is done in isolation. We almost never own the data. people who do own the data have strong views about what we do with it, but also innovators. And some of the work I'm going to show you was actually done by a Google Summer of Code student, and some of it was done by collaborators, and we found a way to bring that into a service model. And then for the value piece, obviously, we cost money. If we've got 390 petabytes of raw storage, that's quite expensive. But we have to look at the value and cost and usage of the data in the context of that cost, the cost benefit of adoption for our integration strategies, because that matters too, and how we align with the scientific drivers community need. And so next I'll talk about some of the challenges we have about scientific drivers and community need. So I'm particularly interested in interoperability for integration. So, and in the world of vocabularies and ontologies. And so many of you are probably familiar with ontologies, but I thought I would give a really quick primer. Most people are familiar with the gene ontology because almost no analyses get done in genomic space without the gene ontology telling us something. And the gene ontology represents the universe of biology, so the functions, locations, and processes that gene products carry out. And so it's not really about genes, it's about gene products. And so the gene ontology is a representation of the important things in some particular space. And in the case of the gene ontology, it's about the kind of entities, genes, and the cells, that's where they exist, or sometimes in the intracellular spaces. Go and other ontologies are an active formal computational artifact, so the mathematical models built on first order logic. And we have lots of tools that can process them. But actually, to make them usable, we really need those tools, and we need the tools in the context of our data. And I'll talk a bit about that. And there's also a kind of common language for communication between collaborators, a shared understanding. And actually, lack of shared understanding is a really common problem in data integration. And it allows data sharing, but it also allows us to produce integrations of data for particular scenarios. So I brought an example of a interoperability problem. So this is a human one. This is dyschromatopsia, or color blindness, which is an abnormality of the eye. It's X-linked, so some of you watching this may not be able to see my little test dots there and to see the green number on them. If you look in the scientific literature, this will be described in lots of different ways, and in any free text that any user sends to any data deposition database to describe their samples, you may see some or all of these words or none of them. And so we spent a lot of time trying to mine things out of the literature and formalize them. And so this is a directed icyclic graph from the human phenotype ontology that shows how we model dyschromatopsia as an abnormality of color vision. It's an ISA, and then we've got some subtypes as well. And this becomes important when we want to traverse the entire disease space, and we can describe cancers in the same way or other genetic diseases. So when I annotate data that comes in through our data deposition resources, it doesn't represent our boundaries of knowledge representation. And people tend to think, I work in this space, I work in cancer, my vocabulary is about cancer, or I work in rare genetic disease, and my vocabulary is about rare genetic disease. But in practice, I work across all of those spaces because we're trying to do data integration across all spaces and then bring it together for particular uses. And so if I look at genome-wide association studies, my group's delivered the genome-wide association catalog for more than 10 years. When we want to describe things that have happened to samples and to describe the knowledge that we get from those samples and also from people, we're talking about traits and measures, anthropometric measures, height, weight, hair color, diseases, response to drugs, response to vaccines is interesting at the moment, activity, diet, blood chemistry, gene function. So all of those things are interesting to us. And so when we want to describe the data, we mash it up into an application ontology that describes our data. And describing our data means that we can do interesting things with it. And so this is the underlying service where we build this ontology. But we also need to be able to apply it. And that's why we have a Semantics as a Service toolkit. So these Semantics as a Service tools allow us to find terms to annotate our data. They allow us to map between ontologies. So if you're going to work in clinical space in Europe, you're going to be dealing with ICD or SNOMED, and those two things need to be mapped together. But if you're working outside clinical space, you might be using a different ontology, or you might be using a domain-specific one, like the Orphanet rare disease representation. So we're interested in finding terms, visualizing them, but also mapping between them, understanding the provenance of the mappings, and cross-referencing between them. And most recently, we've built a data annotation service. So this was written by a Google Summer of Code student that wasn't in my group, but he did something really cool, and we've actually hardened it. And we now got a term request broker, which allows people to annotate their data. So these services get used by every EBI data deposition database, when they acquire data, so that it's acquired in a state that can be interoperable when it arrives in the resource, but then we layer on some additional services to make things interoperable, like cross-ontology mapping. And I will move on to talk about some examples. So this is the NHGRI GWAS diagram. And so in 2011, I got a phone call from Tom Manolio, who is the Division Director Genomics Medicine at NIH. And they had been drawing this beautiful dot diagram, which shows all of the known SNP trait associations derived from GWAS studies since about 2008, and laid onto the human carrier type. That's the chromosomal representation. But what you perhaps don't know is that it was drawn by hand. So this is an image, it's a cover from Nature Genetics, and it's done by the same artist that was drawing this dot diagram. And his name is Darryl Lager. He's a really talented artist, but he was spending a lot of his time drawing these dots because there was no way to automate this. And critically, this is the key for the diagram, and there are more colors here than can be understood by the human eye. So you can't see these colors on the screen very clearly, and you certainly can't perceive them when they're presented in a very dense matrix. And so what my team did was take the GWAS catalog data. So we've got a set of SNPs and traits, very variable traits. There are thousands of traits associated with these variants, which were aligned to build 38 of the human genome consensus sequence. And we color-coded them, and we did this with an ontology, and we were able to produce an interactive diagram, but we were also able to filter it. So in this diagram, I'm looking at metabolic disease and immune system disease, and we can see where the SNPs start to stack up. And here we've got the MHC up here, and you can see we've got a lot of association there. So we've taken the data set, we've integrated it from lots and lots of different studies where the data has been captured from the literature, and now we've made it interoperable by putting together the ontology on the top of it. Now we can really start to do interesting things with it. And as people are now demonstrating that the curated SNPs are not particularly useful, they need them in the context of the entire data sets where we get complete summary statistics of all the SNPs that were tested and those that were imputed, so that we can actually go ahead and start to do some meta-analyses. But still, we need to harmonize those data and integrate the semantics, otherwise we can't determine which are the cases and which are the controls. And the thing that we're integrating right now is UK Biobank and FinGen data, where we've got population scale studies of hundreds of thousands of people, but we still want to be able to mash them up into this integrated view. So we've gone past genome-wide association studies into polygenic scores. And this is an example of where we've taken some of the services. So all of the services I showed you that are semantic services underlie both the GWAS catalog, but also the polygenic score catalog. But it was very clear that the polygenic scores, so where we aggregate the effects of variants into a score, which predicts some genetic predisposition, are going to be useful in the context of clinical data. So we want them to be reproducible, but we also want them to be portable. So we want the pipeline that calculates them to be pushed into other resources. Because if you want to use the whole genome sequence samples in UK Biobank, you have to be able to move your analysis pipeline, but you also want to know that the score is reproducible, and you want to know how many places that particular score has been used. And so we worked with Mike Inoue and the University of Cambridge to extend the infrastructure we built with the GWAS catalog to extend it for polygenic scores. It builds on our infrastructure, our curation processes, and our data acquisition tools. It reuses some of the software, and it leverages innovation that Mike was doing in his group, but that we knew how to harden and how to run on our servers. It's collaborative and it's cost effective because we're building on top of things we already know how to do. And it really reflects the way the field is going. We've also written a grant to work with general practitioners in the UK to have polygenic scores reflected in some clinical records, because we have enough people now who are going to be sequenced quite soon that actually it will make sense to represent this information. And combined with other risk information such as familial risk or exposures, it will make sense to do lifestyle interventions at the point of primary care. And we're involved in a project where they're recruiting individuals into a clinical trial, both in Finland and in Italy, to understand how they can change behaviors for people, for example, who have a high risk score for coronary artery disease. So we're really starting to get very close to clinical care. I'm going to move on a bit to drug discovery. So one of the reasons I mentioned open targets earlier is that we've been working with open targets for the past 7 years to deliver data and also to deliver some services to make the open targets platform work. And so we know that finding targets for drugs requires bioinformatics, and most pharmaceutical companies run some kind of prioritization program, looking at community data sets, but also including their own data sets to prioritize targets and to deliver candidates which will go through their drug discovery process. We've got lots of services at EBI that support drug discovery. We've got the genome services, proteins, and Campbell provides us a small molecule library. There are protein structures. Most recently, we've got predictive protein structures from AlphaFold pathways and also genome-wide association studies. So we know that all of these data sources exist, but they're not mashed up for the drug discovery use case. And this was a really paradigm-changing piece of work for me, because it meant that we could finally get people to adopt the services we've been building and start to think about how to do integration on a really large scale. So this is the Open Targets platform. And I have shown a disease page for this for pulmonary arterial hypertension. And the reason I chose this one is I was involved in the positional cloning of this gene back in the 90s. And what we were doing was looking for this gene in a very large interval. And it took us a good 5 years to find it because we didn't have families with good recombination events in that interval. But now we know by mashing up data from multiple databases that we've got 1,952 targets associated with primary pulmonary hypertension or pulmonary arterial hypertension. It's showing a synonym here. And what we've got is data coming from lots of different data resources. So the association score shows the genes and their association with the disease, but it's calculated from the genetic associations. Those come from the GWAS catalog, the somatic mutations, the drugs that are known and associated pathways, text mining from the literature, gene expression and animal models. And you can see it's kind of patchy where we've got data sources. But all of these data sources correspond to a model around this gene and disease label. And we're using the ontology that we also use for the Polygenic Score catalog and for the GWAS catalog to represent this data, to make it integratable in this scenario. And you can see some of the structure of the ontology here is visualized. And it turns out that the people in pharmaceutical companies think about therapeutic areas. So they want all of their diseases mashed up into therapeutic areas. And that's their most interesting entry point for data. But that isn't, of course, the case for people who are not developing drugs. So we've got an axis here also around known drugs and therapeutic areas. And here's a second slide. And at the bottom of this slide, I've shown the target profile and the evidence piece. Is their known drug data, is their mouse phenotype data, is their pathway data, is their expression data. But actually we can go beyond this now, and we can actually start to look inside individual studies, and the most commonly used ones are large GWAS, so UK Biobank and FinGen are great examples of that. And we can actually start to get past causation, past association and into causation. So we can also start to look at effector genes and starting to figure out which effector genes are present in which cell types. And so we're going to be bringing in data on cell types, we're going to be bringing data into effector genes, and there are multiple sources of these data and multiple views on what's an effector gene in this sense. And so that's sort of the next phase for this, but those are data that we will also be collecting and aggregating. And I should say that almost all of this work was done by the Open Targets team, but we collaborate with them to deliver some of the services, but also some of the critical data sets. So what does good look like? I've talked through some examples and I've talked about how we do it. So I said things need to be scalable, extensible, and modular. And I'm showing how we can scale from one technology, genome-wide association studies, and start to think about application areas. We're also having to scale just in terms of data sets. And now we're actually got really large numbers of summary statistics. We're starting to think about the strategies for making those available, and actually querying them in real time is starting to become really problematic. So we're looking at strategies to do knowledge, some of the summarization of that knowledge, and to pre-compute some of those queries to make sure that they still work. We want things to be modular because we want to reuse the services, but we also want to reuse visualization components. We want things to be simple for users. So everything that I've shown you has its own user interface. So very often we will use a standard set of APIs, but build different interfaces on top of them. So for example, we're using Ensemble as a source of our genomic information in the GWAS catalog, but we use a widget called Lockersoon, which actually comes from Mike Bonke's group in the US, to visualize that data for users. And it's plug and play. So we drop that widget in our infrastructure and it works because we fed it the right integrated information. The quality is aligned to the use. So OpenTargets has very particular quality standards, and they remap everything onto a build of the genome they're happy with, and they use their own mapping strategy. And we've got clear licensing here. So all of the data that comes in as summary statistics to the GWAS catalog comes in as CC0. All of the data that comes into the polygenic score catalog is CC BY. And we are working with the governance models. So it used to be that we recruited this data from the literature, but when people send us summary statistics, some of that data they don't want to reveal to us because it can potentially reveal some information they don't want to reveal about the genomes of the individuals. And sometimes there are traits that they don't want to reveal either because they could be identifying, or they could also be socially sensitive. So for example, we hold some data around sexuality that people don't always want to share. And we can get from the data to the knowledge and the knowledge to data, and we can traverse those pathways reciprocally so we know what the provenance model is. And so when we stack something up and we can say to a pharmaceutical company, we believe that this information is usable for you, that the provenance and where it came from and which data sets it was derived from comes with it, so they can make a decision, but also so they can incorporate their own data. And so they do not incorporate the same data sets that we have, but weight them in different ways, so they're not double counting. And all of this is stacked up in the findable, accessible, interoperable, and reusable space. And the utility of FAIR principles, I think, has been to set us on a journey, particularly about our licensing models. People who operate very large databases have data coming from many, many different sources. It becomes really challenging to retrofit open licenses to those for past data, but we can change how we make the data open in the future and to choose our licenses. And that means that the data will be more highly reused. And indeed, we are seeing where we put open licenses on these data, we're seeing really high usage, particularly of summary statistics data. So I'm going to finish there and just talk really briefly about Outlook. So I think we are going to need cloud portable services, shared technologies for common integration scenarios. Lots of work has come out of Global Alliance for Genomics and Health, and there's a nice paper in Cell Genomics. We need portable analyses. We're shipping off our analyses now, but often we're not doing them in-house. Data readiness for integration and analysis at source. And really we're doing integration as a service on demand at scale, because we have to build more things for more data use cases. And so the interoperability I've talked about here is enabling the integration, but the integration will continue. And I'd like to thank my team. I don't have photographs of all of them because we've had a lot of new people join us in the last year, and it's very hard to take consistent photographs if they're all sitting in their houses. And I want to acknowledge the funders as well that have funded this work, and it's come mainly from grant funding from Europe and from NIH. And with that, I can finish. I hope I haven't gone over time, and I can take any questions.